Hi guys, Miss Francis here. We are starting our next unit, which is all about the cell. We're going to explore the cell organelles, which means the parts that are inside of the cell. So let's start by first defining life. How do we know that something is living versus non-living? Well, scientists categorize something as living if it grows, both in cell number and size, if it can synthesize bigger molecules from smaller molecules, if it does respiration, if it makes ATP, if it is capable of reproducing, if it does nutrition, if it obtains nutrients and vitamins, and finally, does this organism excrete? And does this organism respond to stimuli? All taken together, these processes are known as metabolism. And what an organism that's living tries to do is to maintain homeostasis, where all of those life functions are balanced. So homeostasis involves the collaboration of all of our systems in order to maintain a balanced, stable internal environment where our optimum pH and our optimum temperature are kept. Otherwise, that could impact our chemical reactions. And I'm talking about these terms in relation to human beings, but obviously other organisms are also going to do different processes to keep homeostasis. So how do we know what we know? Well, we make theories and we base those theories on observations which come from facts. So the cell theory has four main components. The first one being that living organisms are composed of cells, where the smallest organisms are unicellular, meaning they have one cell, and the larger organisms are multicellular. They're composed of many cells. All cells come from pre-existing cells. Cells are the basic unit of structure and function, and the cell is the smallest unit of life. However, there are always exceptions to the cell theory. One of the exceptions to the cell theory is striated muscle. Striated muscle is much larger than regular cells, but it's still unicellular. And each muscle fiber contains hundreds of nuclei. Usually each cell only contains one nuclei. The giant algae dispute this theory because the giant algae are only unicellular and have only one nucleus, even though they're rather large. So according to the cell theory, scientists would expect these giant algae to be multicellular. Another exception to this cell rule is aseptate fungi. Aseptate fungi, they're different because usually fungi are made of thread-like, long tube-like structures called the hyphae, which are divided by septa. However, with these types of fungi, the hyphae are not divided into sections. Instead, they are uninterrupted tube-like, containing many nuclei. So if you look here, you'll see the hyphae that are undivided, each containing many nuclei, which disputes the cell theory. That goes along the cell theory. So is the cell theory valid then? Because there are exceptions to the rule, but there are also other parts of the cell theory that seem to be broken. For example, the fact that one of the parts of the cell theory says that cells are the basic unit of structure and function. Well, viruses and prions, which are self-replicating proteins, they can reproduce inside the cells. So then are the viruses and prions the basic unit of structure, or are those cells the basic unit of structure and function? Also, how did the first cell develop, right? If all cells came from pre-existing cells, Well, then where did that pre-existing cell come from? It's kind of like the what came first, the chicken or the egg kind of argument. Regardless, how do we even know that cells exist? Well, it's called microscopes. Microscopes enable us to look at cells in a way that is not available to the naked eye. The first type of microscope that was invented was the light microscope or the compound light microscope. Those are the ones that are in the closet in our classroom, where light is passed through the specimen and then passed through the lenses and magnified. Now, recently, we have been able to increase the magnification and the resolving power of our microscopes, where magnification just means the ratio of the object's image to its real size. So what do I see when I look inside the microscope relative to its actual size? And the resolving power is the measure of image clarity. In other words, what's the minimum distance that 2 points can be separated and still be viewed as 2 separate points? So here is our modern microscopes. 2 types. We've got the scanning electron microscope and the transmission electron microscope. So you got the SEM, the scanning electron microscope, and the TEM, the transmission electron microscope. So the scanning electron microscope gives us 3D surface images, where the transmission electron microscope allows us to view inside the cell and take a look at the organelles. So here we go. The transmission electron microscope used to study the internal ultrastructure of the cells, where in this picture, you're looking at the inside of the small intestine. Here is a transmission electron micrograph of HIV particles, whereas the scanning electron microscopes are going to kind of give us a 3D image. So this image here is the same part of the body, the small intestine, that we saw in the picture of the transmission electron microscope micrograph, whereas here we're looking at the surface of the structures, and it kind of gives us a 3D image. So let's take a look at some other examples, right? Here's the HIV particles budding on a human lymphocyte. This is the scanning electron micrograph picture of it versus the transmission electron micrograph picture that we saw earlier. These are dust particles. I'm sorry, these are viruses. Here is a dust mite. Here is a close-up of, I'm not sure what this is actually, to be honest with you, but here's the eye of a fruit fly. And now check this out. That's actually the surface of the eye of the fruit fly. And these are all 3D images that are produced by that transmission electron microscope. So here's what a scanning electron microscope would look like. And here's what a transmission electron microscope would look like. These microscopes are extremely expensive. They take up a large area, so we don't have these microscopes in our school. You would find those in like research labs or labs in hospitals. There are some positives and negatives to using the electron microscopes, the more recent, more technological microscopes versus the light microscopes. So while the light microscope cannot resolve individual cells, it can resolve much of the internal anatomy. I'm sorry. While it cannot look at the internal anatomy, the organelles, whereas with the electron microscopes, you can look inside the cell and view the organelles. However, this is just another way of saying the same thing. Light microscopes allow us to see the structure of cells, where electron microscopes allow us to see the ultra structure of cells. In other words, what's inside of the cells. But unfortunately, because electron microscopes use electrons to pass through the organism in order to create the image. They can only be used on dead cells, whereas light microscopes only use light. So although they don't have as high a magnification and as high of a resolution as these electron microscopes do, they can be used to study live cells. Cell fractionation helps scientists separate the organelles within the cells, and it's done using a machine called the ultracentrifuge or the centrifuge. Ultracentrifuge just means it's a smaller centrifuge, and this we actually do have in our research classroom at the high school. Basically what it does is it spins really fast, separating whatever is in your test tube based on their density. And this enables scientists to study the individual organelle and learn its individual function. So here are some review questions to see if you really did understand the different types of microscopes and which one should be used in which situation. Next time we'll be comparing prokaryotic cells and eukaryotic cells. See you then.